Okay, morning or afternoon or evening, everybody. Today, so I just wanted to go over this speciation lecture a little bit, and we'll talk briefly about the activity that you should have done in class with Anna, and then we'll cover a few things at the end that are sort of continuing to work through that worksheet that you had. So as you're watching this, make sure you have that handout with those 7 different questions and prompts on there as we go through and talk about speciation. So speciation is one of my favorite topics, and you may have discussed this already a little bit in 112. This is one of my favorite topics because it helps explain why we have so much biodiversity on Earth. So all these different images here, these are very dramatically different lineages of species, and these have all evolved over millions and billions of years because through this process of speciation. And oftentimes, and I like to also talk about this this topic and do this activity, which maybe frustrated some of you, it always does, because it illustrates the fact that in science, not everything is black and white, and we don't have all of the answers. And sometimes there are multiple correct answers, and sometimes really good explanations are not perfect, and they have their flaws, and they don't work in every scenario. And so I hope you've been able to sort of see that as we've gone through this activity. So in class, you should go through this this activity with Anna here, where hopefully at this point you had a discussion about how many species you think there are based on just this image, these 5 images of these different orchid populations. Okay, and I normally get lots of varied feedback here. Some people say, oh, they're all 5 different species. Some people say, oh, they're all the same species, and they just look slightly different. That different color pattern is just sort of normal within species variation. But the point here is to get you to start thinking about what you know about species, what a species is, how you define a species, how you would delineate between different species, so that then we can kind of dive in a bit deeper and figure out what the science says, what the data say. Okay, so you should have gotten each of your group should have gotten a data set to work with, and this was the first data set. And so the goal here was for you to consider, based on this data set, how many species you think there are. Okay, does that influence your original assessment? And then talk with somebody else who has a different data set to see what they thought. And hopefully that went okay. Again, this is an exercise in sort of hopefully seeing that there are different ways of doing things and that there isn't always one clear and clean answer. So this first data set was sort of this morphological data set with different flower sizes, or looking at how many flowers within these populations had different flower sizes. And there are lots of different things you could have come up with here, okay? You could have said, all right, well, these 2, for example, A and D, are different species because they have different average flower sizes, but maybe A is the same as E because they have kind of the same average, and this one's E is just a little more variable. Maybe C is a hybrid. So there's lots of different things that you could have come up with here in different ways that you could have justified that, okay? And so this is kind of representative of what's called the morphological species concept. And this defines species based on structural features, okay? So things like morphology, anatomy, physiology. And there's some advantages and disadvantages, and hopefully you came up with some of these and saw some of these as you were working through this. So some advantages are that it can be applied to sexual and asexual species and fossils, and oftentimes it's easy to characterize. So the morphological species concept, you can go back in time and look at things that have been dead for millions of years and look at their physical characteristics, and that might be really helpful in certain situations. Some disadvantages are that morphology can change over the lifetime of an individual, okay, where it can change between different environments or based on diet. You could have cryptic, sort of hidden species. So in this image here in the bottom right, these are 2 different tadpoles, spadefoot frog tadpoles, and they differ really dramatically. These are siblings, yet they differ dramatically based on their diet. Okay, the one on the right, the really big one, is a carnivore. The one on the left is an omnivore. So that can be kind of misleading if we don't have all the information about those species, about those individuals. Okay, data set 2 looked at pollinators and which population these different pollinators visited. And here, the idea was for you to start thinking about reproductive isolation and sort of gene flow between populations, the potential to interbreed. And this kind of represents the biological species concept. The advantages here are that has the potential to unify lots of other concepts because how whether an individual recognizes another individual as a species, the same species, or a potential mate, or whether they're able to mate, will oftentimes depend on things like morphology, and so this can kind of unify lots of different concepts and represents reality. Some disadvantages are that it can't be applied to things like fossils or asexual organisms because they just clone themselves and divide rather than breeding with other individuals. And sometimes it's hard to define that potential to interbreed. Okay, dataset 3 looked at these genetic strands, and hopefully you did this kind of pairwise comparison and found that A and B had 2 differences, A and C had 1, A and D had 2, and so on and so forth. And here, what you might have been banging your head against the wall over is, how many genetic differences matter? And this was just a short data set, but this is a real question, and this approach is the representative of the phylogenetic species concept, where we're looking for kind of the smallest monophyletic group, the smallest group of individuals that share a common ancestry together, and those are thought to represent species. So some advantages here are that it can apply to sexual or asexual species. It's kind of straightforward to gather the data. You just need some DNA, you just need some tissue, and this can also help identify sort of these hidden or cryptic species. So this is a cool example here on the right of some work where they actually use DNA from crocodile mummies to help identify a cryptic species of Nile crocodiles. Some disadvantages are that it's probably what you struggled with. Where do you draw the line? How many differences represents a real species? And also that DNA is oftentimes limited to living organisms. So we can't always go back to Egyptian mummies and pull DNA. So it only works for sort of living or relatively recently preserved tissue samples. Okay, last one was looking at geographic location, and you might have picked out, if you know a little bit about Hawaii, this is the Big Island here. The weather patterns of the Big Island are similar to Oahu. So you have kind of a wet windward side and a dry leeward side. So maybe you picked up here that there were differences in elevation, maybe differences in rainfall, and maybe this geographical isolation caused 2 things to be different species. So lots of things that you could have considered here. And again, it's not really a right answer necessarily. So this kind of represents the ecological species concept, which focuses on that ecological niche that organisms occupy. And this really emphasizes the role of what's called disruptive selection. So the disruptive selection is where 2 populations are living in different environments, different selective environments, and that can drive them apart from each other. And as they're becoming adapted to these different environments, they're likely to start changing in features that relate to reproduction and mate choice and so forth, and that can lead to reproductive isolation and sort of cause new species to form. So the advantages here, this applies to sexual or asexual species. It also incorporates species interactions, things like competition, which are thought to be major drivers of speciation. Some disadvantages include that those niches, those environments in which a species occurs, can change over the course of the development. Just think of a frog, which lives in an aquatic environment as a tadpole and then can metamorphose into an adult that lives on land. Niches can also overlap, so it's hard to determine to what extent niches need to differ in order for things to be considered different species. And some niches are defined by competing species that may only be present, in the environment. And so, defining one species based on its relationship with another species can be somewhat problematic. Okay, so what I want you guys to think about, if you haven't thought about this already, So these are just 4 of a couple dozen species concepts that people have proposed over the years. So if you just think about that fact that there are over 20 species concepts that people, all of which have been presented with valid data and pretty good arguments and published literature. What do you think about this concept of species? Okay, first of all, going back to this example, and maybe Anna dropped this bomb in class, but this example was all made up, first of all, so there isn't a right answer. And even if this was sort of a real situation, oftentimes we don't know, and the solution that we arrive at, or the answer that we arrive at, very much depends on the approach that we take. And that's the point. That's the point of this whole exercise. And I know many of you want a right answer, and this will be frustrating for you, but I'm okay with that. So if you haven't thought about this already, think about this. What do you think about this concept of a species? Okay. Some of my thoughts are that, well, first of all, this is a helpful thing to think about, because we need to have ways to identify species in order to understand them, in order to study them, in order to protect them. And that's one reason that we try to describe species and define what a species is. But also, this, like many things in science, can be really difficult. There's this big gray area of when what a species is, when something becomes a new species, what defines a new species, and it's okay to feel a little bit uncomfortable about that, and uneasy about that, and that's how you should feel, and that shouldn't make you doubt science. That should make you confident in that scientific process, because that uncertainty leads to further research and leads to further inquiry that can help us better understand things to the best of our ability. Okay, so I like to think of species kind of like colors. Okay, so nobody would argue that blue is not a color, or that yellow is not a color, or that red is not a color. But if you really look at what these things are, what these colors are, these discrete categories of colors, these are just a point along this electromagnetic spectrum, okay, this radiation that is light, visible light and also invisible light to us. So blue just falls somewhere. It's just kind of a point along this spectrum, this continuum. So is yellow, and so is red. So although it's clear that blue is a color, and it's a distinct color, where does blue become violet, and where does blue become green? That is a little harder to define. And species are very much the same way. Speciation is thought by many, including myself, to be a continuum. It's a continuous process. You're not just a species like that overnight. In some cases, yes, that does happen. Well, not overnight necessarily, but very quickly you do get this very distinctive speciation that occurs. But in many cases, that species boundary is porous, okay, and that is there's sort of these gray areas. Even though we might recognize two things as distinctive species, they might not follow all the rules, and that's okay. All right, that is a, you can consider that to be sort of a fact of species, is that they're not always black and white, and sometimes that boundary is porous, and that there is this sort of continuum of speciation, of reproductive isolation. All right, so we're going to focus most of the rest of the time, and you probably didn't get to this in class, we're going to focus on this biological species concept, okay? And that's because this is really helpful in thinking about reproductive isolation, because that is how sort of speciation occurs and unfolds. And so this biological species concept defines a species as a group of actually potentially interbreeding natural populations that are reproductively isolated from other such groups. So we're going to focus in a lot on that reproductive isolation for the rest of the time. Before we do that, I wanted to talk briefly about the geography of speciation. So speciation can occur in either the presence or the absence of physical barriers between populations that are starting to turn into different species. So when there is a physical barrier, like here on the left, uh, this is referred to as allopatric speciation. Now, when there is not a physical barrier, this is referred to as sympatric. Okay, so sympatric means kind of same geography. Allopatric means different geography. So presence or absence of barriers. So we'll talk briefly here about allopatric speciation before we focus, the rest of our time on some examples of sympatric speciation. So we're in Hawaii, so let's talk about Hawaii. If you're not familiar with this, you should be. I'm making you familiar with it right now. So the Hawaiian island chain formed because we sit on top of what's called the Pacific Plate, which is a big tectonic plate. So if you don't know this, you should know this. The entire Earth is covered in these big plates, these big tectonic plates that are constantly shifting and moving, and that's where our continents are moving, and this is why we have earthquakes, and this is why we have volcanoes and so forth. So sometimes volcanoes occur where two plates come together, but there are also places in the middle of a plate where you'll get what's called a hot spot, and that's where the mantle from underneath that plate emerges through sort of a crack, a little hole in that plate, and that mantle comes out in the form of lava that hardens, and then you get islands. Okay, so there are lots of volcanic islands around the Earth. Some of them are hotspot volcanic islands. So our island chain is a hotspot volcanic island chain. So the reason that this chain is formed this way is because that plate that we're on is constantly moving slowly, very slowly to the northwest. Okay. So as that plate is moving, this crack bubbles up, this mantle, the liquid magma bubbles up, forms an island. That island raises up, gets pretty big, and then that island slowly drifts away from that hot spot as that plate is moving. So it moves in this direction, and then a new island starts to form, and then a new island starts to form, and a new island starts to form, and these islands start to erode, and they start to cool, and they start to shrink, and sometimes they start to disappear under the ocean. So you can actually, I encourage you to get on Google Maps, Google Earth, and look at the satellite view of Hawaii. And if you look to the northwest, you've got the northwestern Hawaiian Islands, which are sort of remnants of once great islands. And then you can kind of follow that underground island chain all the way up to Alaska. And that traces about 60 million years of history. So the current islands right now are about 5 million years old at their oldest. And so as you move away from that hotspot, which is currently under the big island of Hawaii, as you move away from that hotspot towards these older islands of Kauai and Ihau, they become older and older and older. Okay, so these are kind of the time ranges. So this is cool because over time, species have gotten to these islands, and then as new islands have popped up, they've migrated to those islands, and they've settled there, and they become isolated. So this is a classic example of this. So this is these are some fruit flies that once upon a time colonized Oahu or something even older, and then they became a new species, and then some moved over to Molokai, and then they turned into different species as they became geographically isolated, and then some moved to Maui, and then some moved to Kona, and then some moved over to Hilo. So this is an example of this allopatric speciation. Okay, of speciation occurring when there's a physical barrier between those different species, and that speciation can occur either by random chance, because by random due to sort of random mutations, or the nature of mutations. You've got different genetic mutations that are arising in these different populations, and over time those mutations could cause two populations to become distinct enough to become new species. Or you could have these islands maybe that have different environments, different ecologies, and that selects for different, this causes sort of divergent or disruptive selection, and that can cause reproductive isolation between those different species. Okay, so allopatric speciation, we can think of this as gene flow being restricted between geographic isolated populations due to those physical barriers. And then once those populations are isolated, that reproductive isolation, RI,
Could arise via natural or sexual selection,
or genetic drift, so that's kind of random fluctuations in that gene frequencies within those populations, or mutation. Okay, so at this point I want you to take a minute, pause, catch your breath, and summarize, think about and summarize this concept of allopatric speciation, and make a note of any questions you have. You can ask those questions to a friend, Anna, or myself. Take a minute, I'm going to get a drink. Okay, so if you haven't jotted down some ideas here, pause before we move on. All right, so the rest of the time we're going to talk about reproductive isolation and focus on sort of a sympatric speciation scenario. So sympatric speciation is when reproductive barriers isolate a subset of a population in the absence of a physical barrier. So this is when, for example, you've got two fish species that are living within a stream, the same stream, and there isn't anything that prevents them from interacting with each other, from crossing over and sort of mingling with the other population. Okay, so we're going to use as an example, this is some of my research that I did when I was a PhD student down in Panama, Costa Rica, on brachyraphis fishes. So these are live-bearing fish, so they have internal fertilization, just like mammals do. So many fish will spawn externally, so the female lays her eggs, and then the male fertilizes the eggs outside the female's body. That's not the case here. So these have internal fertilization, and they give birth to live young. So they have little babies that come swimming out of the mom after she's been pregnant for about a month. So males display, and then females choose. So these are both males that are pictured here. So they have these kind of bright color patterns on their fins, and also you can see, so their anal fin, which is kind of that bottom fin, that lower fin at the middle of their body. In this species on the right, and you'll see this, we'll talk about these two species in a minute, but this one has this kind of dark color pattern, this one doesn't really have any color on it. So males display and females choose. Okay, so males might mate with lots of different females, but females are picky and they only choose the best potential mates. And they use those physical characteristics to select a mate. So they are both, they're all diurnal, so they're active during the day, and they breed year-round. So they live in the tropics, they don't have a distinctive breeding season, they breed all year round. Okay, so the two different species that we're going to consider here are Brachyraphis terbensis and Brachyraphis roseni. So terbensis is on the left, and roseni is on the right. And these both live in the same streams, but they live in slightly different habitats within those streams. So terbensis on the left, they live in these portions of the stream that are kind of higher flow. They have faster currents. The water tends to be cooler and more oxygen rich. There are fewer predators, and they tend to kind of eat in the water column. So they kind of swim in that current, and they pick off insects and so forth that fall into the water and that then get kind of swept down in that current. Roseni on the right, and then terbensis does not have a pigmented anal fin. Roseni on the right, they live in kind of these backwater parts of the stream, but again, there's no physical barrier between these two parts of the stream. So they live in this kind of low flow parts of the stream. The water is warm. It tends to have less oxygen. It has more sort of rotting debris on the bottom, at the substrate at the bottom of the stream. There are lots of predators, and they tend to eat at the surface of the water. So they swim around and they pick off ants that fall from the canopy and then are kind of stuck at the surface of the water. So with this information, what I want you to do, if you're working with your group, or if you're working by yourself, work on number 5 in that handout, that same speciation handout. So this This says, you've got two species of brachyfish fishes that live in close proximity, Based on the information provided on the slide, what factors or mechanisms might prevent these species from breeding and thus allow them to remain reproductively isolated? Okay, so take a minute, talk about that. Okay, we're going to move on, so pause and take some time to think about that if you have not already. All right, so some ideas that you could have come up with here are that, maybe because they live in different environments, different ecologies, that one species, if it swims over into the other environment, it might not do so well, okay? So if you're Terabensis and you swim over into Rosenei habitat, you might not know how to avoid predators, okay? So you might be eaten by predators more often. If you're Rosenei and you swim out into the high flow, cool water, you might not be well adapted, well suited for that sort of high flow environment. You might not be well suited for eating in that area. You might also think about that idea of mate choice that we talked about, okay? So that first one that we just talked about, that would be called kind of habitat isolation or immigrant inviability. If you move into a new environment, you have reduced survival, reduced fitness, then that could cause reproductive isolation. You might also have sexual isolation going on here, behavioral isolation. So if you're a Terabensis and you swim over into Rosenei habitat, you might not have what it takes to find a mate there. Maybe those females really like that pigment and they sort of cue in on that, and they might know that you're kind of terrible at avoiding predators and they don't want to have babies with you because then your babies are going to be terrible at avoiding predators. So that is called sexual isolation or behavioral isolation, where that mate choice, mate preference reduces contact or sort of mate choice between those different species. Some other ones that are really important in other situations, but that probably don't apply here, are temporal isolation. So this is when populations or different species might not ever run into each other because they breed at different times. They might breed at different seasons, different times of day, or even in different years, like these periodical cicadas, which emerge either every 13 years or every 17 years to mate. And so if you're on a different time schedule, you might not ever run into each other. In this case with these fish, that's not an issue, but it is an issue in many other populations. Mechanical isolation is another one that you might have thought of, and this is this idea that there are morphological, or there could be morphological differences that prevent successful mating even when they try to mate, even when none of those other barriers prevent them from trying to mate with each other. You might have sort of a mismatch in physiology and the physical characteristics of those individuals. So in this case, with these species, males use fertilized females using the gonopodium, so that's that modified anal fin, and the tips of these anal fins are really, really varied in these different species. And so it's kind of like a lock and key type scenario where the gonopodium matches the physical characteristics, the anatomy of the female, kind of like a lock and key. So if you're a different species and you try to mate, things might not work out. Gametic isolation is another one that could play a role, and this is when the sperm of one species can't successfully fertilize the eggs of another. There's a really cool example here in Hawaiian waters where you have different sea urchin species that spawn oftentimes in the same time, the same place, but the sperm don't adhere to the eggs of other species because you have sort of incompatibility in these surface proteins, and that's a good thing. Okay, that keeps those species separate. So all of these different barriers that we've talked about so far, this habitat, temporal, behavioral, mechanical, and gametic barriers, are what are referred to. These all prevent fertilization from occurring by preventing mating attempts, or they prevent successful mating or fertilization. These are called prezygotic barriers because they occur before a zygote forms. Okay, prezygotic. So there are also barriers that could prevent fish from gene flow between two different populations, even when none of those prezygotic barriers are present. Okay, so let's suppose these fish do attempt to mate and they do successfully fertilize with each other. what post-zygotic barriers, or barriers that occur after mating has taken place, what do you think, what kind of barriers do you think could help maintain reproductive isolation? So this is number 6 on your page. So think about that for a minute. Pause it, think, and talk about this idea. What could happen after mating has already taken place and fertilization has occurred that could prevent gene flow? Okay, we move on, so pause if you haven't thought about this already. Okay, so these barriers are called post-zygotic barriers. There's a few different ones. One, you could have reduced hybrid viability. So you might not have used these exact words, but you might have thought something like this, that maybe these hybrids form, but they're just not as good as the purebred. And there's something wrong with them. Either they're not well fit for their environment, or maybe they're not as attractive to mates, and that could cause problems, and that could prevent gene flow, prevent that mating from occurring. You could also have reduced hybrid fertility. So maybe you successfully form this nice healthy hybrid, but that hybrid is either sterile or has lower fertility. So you could think of a mule, for example. So if you have a donkey and a horse that mate, they form this nice, strong, useful mule, but that mule is sterile and it's unable to mate, it's unable to pass on its offspring. You could also have what's called hybrid breakdown, and this is where you have successful, healthy, and fertile hybrids, but then when those hybrids mate with each other, or when they mate with the parents, that reveals genetic incompatibilities that cause those hybrids to sort of break down and to be inviable. So all of these help prevent the formation of viable, fertile offspring. And if any of these is in place, you don't have to have all these barriers, but if one of these barriers is really strong, that can prevent gene flow from occurring, and that can keep species isolated or drive species towards isolation. So I like to think of these as sort of hurdles. Okay, so all of these different barriers have to be cleared in order for them to get to that finish line, to produce viable, fertile offspring. And if they can produce viable, fertile offspring, but according to that biological species concept, we would consider them to be the same species. And obviously, there's some problems there, because there are species, different species, things that are quite obviously by every other measure different species, but they can form viable hybrids. Okay, and that's it's not uncommon, but that certainly doesn't go along necessarily with this idea of biological species. But in many cases, these barriers are really helpful to think about because they help us study this process of speciation and how that isolation occurs. Okay, so last little question here on your handout, which I believe, let me have a look, is number 7. So I want you to think about different possible outcomes of hybridization. Okay, so what do you think would happen if hybrids were as fit or more fit than pure offspring and their parents? So think about this. If two species came together, they formed hybrids, and those hybrids were better than the purebred offspring, or they were even better than the parents, what do you think would happen over time? Pause this, take a minute to think about it. Okay, we're moving on. It's positive if you haven't thought about this already. So one thing that could happen is what's called fusion or collapse. Okay, and this is where different species sort of collapse into one. So if there's no barrier that's preventing them from mating anymore, then they can just become one species. And sometimes those barriers that are preventing species from coming together can be removed. Okay, so this is an example from Rift Lake cichlids in Africa. So these are two different species on the left, and they were only isolated by behavioral patterns. Okay, so they just looked different, and so they preferred not to mate with each other because they had different sort of distinctive behavioral mating preferences. And those are all visual. And so when those waters became more turbid, when they became less clear, that behavioral isolation was removed, and all of a sudden those species, and this has happened with lots of different species, they've started to collapse. Okay, that single barrier that prevented them from mating disappeared, and then those species collapsed or fused. And sometimes that means that you go from having two species to having one species. And in some cases, you have sort of hybrid species formation, and this is actually thought to be really important in several lineages. So you might have two species that form viable hybrids, and that hybrid could become a third species. So that could be sort of a catalyst for the formation of new species. So that's kind of cool. Okay, finally, what would happen? What do you think would happen if hybrids are less fit than the pure offspring and their parents? So in other words, what would happen if hybridization has a high cost? If mating with another species just sucks? What do you think would happen? If your offspring suck, what do you think would happen? Take a minute, pause, and think about that. Okay, pause if you haven't done so already. We're moving on. All right. So if hybrids are less fit, the thought is that strong pre-zygotic isolation should evolve to prevent that costly hybridization from ever occurring. Okay, and this is a process called reinforcement. Okay, and all right, so let's just think about this for a second. I can't remember if your handout has more information on this or not. I don't think it does. So if we assume that the decision of whom to mate with is genetically based, okay, so if there's some gene that says, all right, mate with this guy or mate with that guy, if you have a gene that says mate with that other species, that's okay, that's a great idea, let's mate with that other species. And then if you do that and hybridization is costly, what is going to happen to your offspring? Okay, if you mate with that hybrid and you have that gene that says, yeah, mating with another species is a great idea. If you mate with that other species and your offspring are terrible, then that gene that you have that caused you to mate with that other species, that gene is not going to go anywhere. Okay, you're not going to pass it on to your offspring because your offspring are terrible. They're either dying at birth or they're dying shortly after, or they're mutated in some way, or they're ugly and nobody wants to mate with them. Anyway, they're terrible, and that gene that they now possess is not going to go anywhere. So what that will cause is for any gene, any trait that prevents that hybridization from occurring, that will become more and more common because it will be favored. So if you have a gene that says, whoa, 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 I don't want to mate with that other individual, that other species looks crazy, I don't want to mate with them, or if you have a gene that causes you to mate in a slightly different time or a slightly different place, and it prevents you from hybridizing, then your gene is going to be favored. That's going to get passed on, and over time, that reproductive isolation is going to be reinforced. Okay, and that's a pretty important process. And we could talk more about some examples of that in the future. Okay, so why does all this matter? I like to just throw in a couple scriptures here at the end. From Genesis, and Adam gave names to all cattle and all the fowl of the air and every beast of the field. So he cared about speciation. Okay, and that's one reason we study speciation, is so we can understand how species form, and so we can identify those species and take care of them. From D&C 104, for it is expedient that I, the Lord, should make every man accountable as a steward over earthly blessings, which I have made and prepared for my creatures. I, the Lord, stretched out the heavens and built the earth, my very handiwork, and all things therein are mine, and it is my purpose to provide for my saints, for all things are mine. Okay, so these things, if we believe these things are Heavenly Father's, and we should care for them, and we should be interested in them and how they form and how this process happens. And I don't think all this happened by, sort of the flick of a switch, the flip of a switch, or, that God snapped his fingers and that's how we get all these millions of species. So understanding that process, I believe, helps us sort of get a glimpse into how that happened, into how God worked that, if that is how we're viewing that thing, okay? And I don't think it's it's one way or another. I don't think we have to view this as, God created all these different species and he did it like that, or this is a scientific thing. Okay, I don't think that's the case. I think these two things could be reconciled And I think Heavenly Father wants us to understand those things. Okay, so if you've got any questions, let me know. Send me an email or talk to me in class or talk to Anna. Hopefully this all makes sense, and hopefully you're all excited about these things. And I will see you guys on Monday when I get back, and we'll keep cruising through the material like we need to. Okay, good luck. Have a good weekend. Stay safe.